Hi, thanks for the invitation. I'm going to share my presentation. Yes, should be okay, right?
Yes, that looks great.
Okay, so we will talk about desert cyanobacteria when they are exposed to non-Earth condition. So I'm going to talk about Mars, of course, icy moon, and also planets that are around other stars. And these three are actually the topic of astrobiology research because they are they are potential habitable either in the past, talking about Mars, or they might be currently habitable, like the icy moon, or also exoplanets that are orbiting around M-star. And diciamo, to look for life elsewhere, in my laboratory we use desert cyanobacteria as a model system because they have some special feature that make them so suitable to test the limit of life under non-Earth conditions. So first of all, I want to say that I'm going to talk about cyanobacteria that are belonging to the genus Crocosidiopsis. And these cyanobacteria are used to live in the most extreme environment on Earth, like hot and cold desert. And in these places, the conditions are so extreme that they can survive only taking refuge inside the rock. So epilitic community on the top of the rock are not present in these extremely arid places, but you can find cyanobacteria inside the rock, but also in the space between the rock and the soil. And I have a collection of cyanobacteria of the genus Crocosidiopsis that has been collected by Imre Friedman and his wife during their lifetime expedition in a desert like the Gobi Desert in Mongolia, the Negev Desert in Israel, or the hyperarid core of the Atacama Desert in Chile, or the dry valleys in Antarctica that are quite famous, and I will say later on why, and also sample that has been collected from the Mojave Desert in California. So the most famous desert strain of Crocosidiopsis are those that has been isolated a long time ago, back in 1976, in the Dry Valley in Antarctica, at the time when the Viking mission failed to give evidence on the presence of life on the Martian surface. We know that the Vikings made some experiment using the soil and looking for microbes, just adding growth medium, and they check for evidence for some kind of metabolism. And then they also tested the presence of organic molecules through gas chromatography, and they found no evidence of life. And since life as we know it is based on carbon, this was very much disappointed. So at that time, Friedman and his wife, Roselio Campo, published in Science the discovery of this community inside the rock in the McMurdo Dry Valley that were considered without life since then. And they also ended their publication saying that in order to look for life and to find life, sign of life on Mars, we should look not on the surface but under the surface. And indeed, now we are looking for sign of past life on Mars, just looking under the surface. Perseverance is collecting sample at 8 centimeter deep under the surface, and the European mission Rosalind Franklin that will be launched in 2028 will look for life, for signature of life, drilling at 2 meters of distance from the surface. So desert cyanobacteria has been considered very interesting to look for studying the limit of life in dried environments. And their discovery is still mentioned in the NASA astrobiology, I mean the NASA graphic novels, especially in the issue that is dedicated on astrobiology and life in extreme environments of Earth. And indeed, if we look at places like the Atacama Desert in Chile, and then we look at the image that the Curiosity rover is sending to us, it, the scenario is really very similar. You cannot say where you are. And so for this desert cyanobacteria, desert environment and life form there are considered to give us an insight on what could have been happened on Mars when the surface, the condition of habitability on the surface disappeared. Because when we talk about habitability, we consider the place habitable if there is liquid water. And in the past, Mars had the liquid water on the surface, then that disappeared. And so life form, if present there, could have taken refuge inside rock, like it does on Earth. And before exposing desert cyanobacteria to non-Earth conditions, so either in space or using planetary simulation that are available on Earth, we really should know how these cyanobacteria can face desiccation on Earth. And so already when I was a PhD student long time ago, we actually pointed out by staining the cell envelope of the desert cyanobacteria that they are very rich of isopolysaccharides. And these isopolysaccharides are really helping the cell to lose and the water very slowly in order to put in place all the biochemical strategies to face the water loss that otherwise will be lethal if the organism doesn't put in place strategies to avoid all the damages that water removal will induce. And also these desert cyanobacteria are also very interesting because they are unicellular, but they undergo binary and multiple fission division, so that inside the multicellular aggregate, some cell die and others survive. So this is also very interesting. It is like that survivors can eat and take advantage of the dead cell, and then when you put them back to life, so you rehydrated this dried sample, they go back to life and they start to make a lot of division. They all the isopolysaccharides that they have been accumulated while dry, they are removed, and they form this big multicellular aggregate, and they are so going back to life. And so if you look at the DNA damage using, let's say, a method that is going to look in situ, highlight the presence of DNA damage, you can see that there are cells with a lot of DNA fragmentation. So you have a lot of this fluorescence dot-like signal, whereas other cells maintain, let's say, a background signal. And these survivals are also related to the permanence of photosynthetic pigments, whereas those that you can see, they have a lot of dot signal highlighting the presence of DNA fragmentation, they don't have photosynthetic pigments anymore. So there is really this selection. I mean, at the moment, at the onset of desiccation, you have a cell that maybe are in a different metabolic state, and so that some cell can survive and other will die. And so you see also with other technique like this staining for life and dead cell, that you see these are dead cell, so the staining can enter and you don't see the DNA stained with another fluorochrome, whereas you see a live cell where the staining cannot enter and the DNA is still there. So we actually um, uh, prove, we investigated this dried cell of Crocosidiopsis with different approach, and we had different evidence that actually there are cells that are able to limit the DNA damage. And of course, surviving desiccation means also that you have to stabilize your subcellular structure while the water is removed. And there is the trehalose, I mean, the water replacement theory that is very well diffused among organisms that are named anhydrobiotic. And in this case, it is very well known that trehalose, but also sucrose, can replace the water molecules, and so that they can avoid the phase transition of the lipid membrane, so that they remain in this liquid crystalline state that is the normal one. So when water is added and they are hydrated, there is no leakage, and so there is survival. Whereas most of the organisms don't have these strategies. And so when desiccation is arriving, there is a transition of phase of the membrane. And when there is the radiation, the following radiation, there will be a leakage of solutes and cell death. Since cyanobacteria are photosynthetic one, they produce also sucrose that is as efficient as trehalose in doing this protection. And when we looked at the genome, in fact, we found all the pathway, all the possible pathway to produce sucrose and trehalose using different substrate, so that they can make trehalose using whatever is present basically, and they produce sucrose through the photosynthetic activity. So this is also a way through which they can survive desiccation. And the other strategies is to protect protein against oxidation. So we investigated the carbonylation of the protein. So we extracted all the protein from dried cell. And as a control, we used the Synecocystis PCC 6803 that is very desiccation susceptible. And in fact, it dies when you dry down very quickly. After 1 year here, over inside the over the silica gel, there were no survival, from Synecocystis, whereas Crocosidiopsis desert strain, we gain a lot of survivals, and this survival is correlated with the absence of carbonylation, of protein carbonylation. So if you look in the liquid state of both cyanobacteria, we have some background level of carbonylation, because anyway oxidative stress is present in this photosynthetic organism. But when we did the same after desiccation, we see that in Crocosidiopsis there is no so an evident increase of carbonylation like we can see in Synecocystis. So this means that during the recovery, when you need actually protein also to repair damage, so we are thinking about also DNA repair proteins, they are still active, and they because they didn't undergo extensive oxidative damage. And indeed, if we look, when we looked at the genome of Crocosidiopsis, one strain of Crocosidiopsis, and we made a comparison with Synecocystis, we figure out that there are a lot of genes, and there is really an enrichment of genes that are encoding for superoxidodismutase, catalase, peroxidase. So desiccation tolerant cyanobacteria really a great repertories of genes to say involved in the oxidative stress defense. So this is another strategies to cope with desiccation, because of course at the onset of desiccation there is an impairment of the metabolic function, and you have an increase of reactive oxygen species. So it is good to be able to keep the production of ROS very as low as possible. And then surviving desiccation in desert. Of course, these cyanobacteria are living inside the rock where actually UV is not penetrating, but it could be that they when the conditions are not so extreme, they can maybe go from inside the rock outside in the epilitic part, or they can be also transported by wind from one rock to another one. So having the possibility to protect themselves against UV by producing UV pigment, UV absorbed pigment is also very important. In general, cyanobacteria have two kinds of this pigment, the mycosporin-like that are actually cytoplasmic UV absorbing pigment, and scytonemin that are on the other hand, accumulated in the envelope. And we looked at the genome of, here you see three desert strains of Crocosidiopsis, and we figure out that all three of them have all the gene for the synthesis of scytonemin, and one of them has also the genes for mycosporin synthesis. And the Nostoc puntiforme has been used as a reference because it is a very well-known cyanobacterium able to produce these UV pigments. And this is very important because then if you are exposed to high radiation and you are also dried, this pigment provide the cyanobacteria with a passive protection, and so this is another plus in surviving extreme condition, and it is also a plus to survive non-Earth condition. That is also where I'm going to lead you in a few minutes. And as we know, when you have desiccation tolerance cyanobacterium or desiccation tolerant organism in senso lato, usually they are also radiation tolerance, because radiation is inducing damage either in a direct way, and so of course there is no way to escape this, but radiation is also inducing damage through the production of oxygen species. And so, since, as I said, the desiccation tolerant cyanobacteria are particularly resistant to desiccation, and they also keep very low the production of ROS species, they are generally also very resistant to radiation. So here you see the genome of a desert cyanobacterium. Here you see the control without radiation. Here you see the fragmentation of the genome after exposure to 5 kilogram gamma rays. This is a huge dose that never appeared on Earth. So the selection for this kind of radioresistant is not coming from the Darwinian concept, but it is really a bioproduct of desiccation. And then if you have the recovery, and so you put the cyanobacteria to recover, you see that the genome is built back, and so all the fragments are repaired. And this is possible just also because the protein are not carbonylated, and so all the protein involved in DNA repair can work. And here you see a comparison between 4 different Crocosidiopsis strains from different deserts. I can say here from Antarctica, from Mexico, from Sinai, from Egypt, from Negev in Israel. And you see a comparison with the Synechococcus that, like Synecocystis, is very susceptible to desiccation, to radiation and desiccation, and E. coli, that is also a bacterium that is very much desiccation and radiation resistant, susceptible. And in fact, both of them are very low tolerance towards radiation, whereas desert strain of Crocosidiopsis are very much resistant up to, here you see 15 kilograms. This is a huge dose that only bacteria like the Nococcus radiodurans can face. So here you see the top 10 of radioresistance, and you see that Crocosidiopsis is really standing alone as a photosynthetic bacterium among bacteria that are not photosynthetic. And also UVC, it is also, we tested also the radioresistant of this desert strain towards UVC. Here you see the dose that is very high, 13 kilograms, it's very kilojoule. It's very high. And E. coli is also much susceptible towards coccidiosis, and also the DNA is damaged by UVC radiation, and so survival has to be related to the capability of this cyanobacterium to repair damage. Here you see that we use DNA extracted from cyanobacterium exposed to this increasing UVC dose, and we use this genomic DNA as a template for PCR fingerprinting. And if the DNA is damaged, you don't have the amplification. And so here you see that bands amplicons are disappearing as the UVC dose is increasing. But we are still in the survival limit of coccidiosis. Here you see it's 4.5 kilo joule, and but we can arrive up to 13 kilojoule per meter square. So they are extremely radiation resistant, and all these features, so the capability to survive desiccation, combined to the capability to face radiation, had made these desert cyanobacteria a suitable model for first experiment, astrobiology experiment that were performed outside the International Space Station. So it has been possible to use the EXPOSE facility that is installed outside, and as the name says, this facility allows the exposure of a selection of extremophile in the dried state to space conditions, so vacuum radiation, either UV radiation, so solar radiation, either cosmic radiation, so ionizing radiation, and vacuum. Vacuum is terrible. If you are not able to face desiccation, you will die immediately under space vacuum. And then this facility, if they can, let's say, um, prepare to simulate Mars condition, because we can put inside on Earth CO2, so like mimicking Martian atmosphere at the Martian pressure, so 600, 800 Pascal. Then we use a filter, sorry, to let the UV light to pass only from 2000 nanometer on, so to mimic the irradiation that is reaching on Martian surface, because there is no ozone, whereas on Earth we don't have UVC and UVB because we have the ozone, while in the interstellar, in the extraterrestrial environment, of course, there is no ozone, so we have the full UV spectrum, and the UVC it's a very damaging one because it is absorbed by the DNA, and it also then producing radio releases, and so oxidative stress and so on. So it's very, as I've showed before, UVC is also very lethal to survival. And in this kind of experiment that were performed outside the International Space Station, this has been an experiment performed by now 10 years ago. So the cell were launched to the ISS, and during an extravehicular activity of the two astronauts, the facility was installed outside the ISS. They were kept there for 1 year and a half, so a very long time, and then they returned back to Earth in June of 10 years ago, under the seat of Commander Tim Peake, and these are two of the three expose part that were recovered on Earth. Inside this facility, there were also crocodiopsis samples. And what we did, so first we dried the samples. Why? Because space vacuum will induce desiccation, so the sample has been pre-dried on Earth, and also because Martian low pressure will induce desiccation. Then they were exposed in space, under space and Mars-like condition for 1 year and a half. And in this metabolic state, they just accumulated damage. And when they come back to Earth, we First of all, we examined the dried exposed cell for pigments, for DNA, for carotenoids, so for biomarkers that can be actually identified by Rosalind Franklin rover when it will do the analysis on Mars. And even if since as I said, the Rosalind Franklin will drill the Martian surface at 2 meters. So of course we don't think to find the photosynthetic organisms there. But nevertheless, DNA is the same. Carotenoids can be present also in non-photosynthetic organisms. And of course, we don't expect nothing about photosynthetic pigments. But this is to say that these desert cyanobacteria are able to stabilize their component and that their component can be still detected after prolonged period of exposure under Mars-like simulated condition in low Earth orbit. And what we did also with a small fragment of space exposed cell, we put this fragment, we radiated this fragment, so we checked, of course, for also survival, but we derived a culture and we extracted the genome from this culture and we sequenced the genome and we made a comparison with the reference that has been maintained in nice condition in our laboratory on Earth. And we saw that the genome sequence of the space-derived strain had really the same mutation as the ground reference, meaning that the accumulated damage were actually repaired properly and there was no increase of the national rate of mutation that you can find by comparing independent sample of your reference strain. And a follow-up of this experiment is coming in 2 years, since this desert cyanobacteria will be implemented inside the BioRider. The BioRider is a new vehicle, space vehicle without a crew that is under development by the European Space Agency, and it will be launched by Ariane 6. Probably it will stay in low orbit for 3 months, and then it will be recovered on Earth thanks to the presence of parachute. And what the experiment that we proposed and that has been also selected has been based on the past knowledge on the experiment performed outside the ISS. We will dry cyanobacteria on Earth. We will expose them to ionizing radiation. Then they will be integrated into a payload that actually it is a miniaturized photobioreactor, so that we can perform the in-orbit rehydration and allow DNA repair to occur under microgravity, because what we have seen before was the repair under Earth conditions. So now we want to see the effect of microgravity and also the effect of space radiation while the cyanobacterium is repaired, induced DNA damage. Then the sample will be fixed and they will be returned to Earth, and then we will make the genome sequencing to see how the absence of gravity will interfere with the capability of repair DNA. And this is also very important because it is in the roadmap, let's say, to use this cyanobacteria also for space biotechnological application. And I will say something more later on. So, being able to survive desiccation, UV radiation, ionizing radiation, these cyanobacteria, as I said, are a nice model to see what happened on Mars when it change from being warm and wet to the dry and cold desert that is now. And we can understand how life could have faced this transition using Mars simulation chamber that we have. For example, we use the one that is present at the DLR in Berlin, in Germany, so the German Aerospace Center, and we simulated for 3 days the Martian condition, so temperature up and down the freezing point, low pressure. So what is happening is that the low pressure will induce desiccation. So also the photosynthesis will quickly disappear. But cyanobacteria, when we rehydrated back after that we recovered them from the Mars simulation, they were still alive. And among the desert cyanobacteria that I have in the lab, there are some that are able to perform photosynthesis beyond the visible light. So, as I wrote here, cyanobacteria capable of far-red photosynthesis are rare because in order to find just two strains, one from the Negev desert and one from the Utah desert, we really screened the more than 40 strains from the collection. So most of them are not able to do that. And this is because the light that is passing through different kind of rocks is different depending on the rock composition. And in some cases, some rocks are filtering the solar light and allow only infrared light to pass through. So in nature, there are cyanobacteria that are capable to transform chlorophyll A in chlorophyll F, as you see here. So we grow this under visible light, and so this is the spectrum, the absorbance spectrum under visible light. But if you grow cyanobacteria with the far-red light that is around 530 nanometer, then you have the transformation of chlorophyll A in chlorophyll F, and this chlorophyll is able to use visible light beyond the visible light. Of course, to do this, you need an enzyme that is going to transform chlorophyll F and chlorophyll A in chlorophyll F. You need also to make a remodeling of the photosynthetic apparatus. So Crocosidiopsis, these two strains have a cluster of genes that codify for paralogs of protein that are able to also the antenna apparatus that are able to use the infrared light. And compared to Crocosidiopsis thermalis, which is a strain that has been isolated not from the desert but from soil in Germany, this Crocosidiopsis thermalis strains has a far lip cluster of 20 genes, whereas the desert strain of Crocosidiopsis has a reduced cluster, has a reduced number of genes. In fact, they modify mostly photosystem 1 and not the photosystem 2. And they can do, even with this reduced remodeling of the photosynthetic apparatus, they are also efficient in using far-red light. But why are we interested into this? Because in the context of astrobiology, there are in our galaxy, there are a lot of planets in the habitable zone of their star, and these stars are M stars. And these stars have an emission spectrum that you can see here that is very different from the emission spectrum of our sun, because our sun has a strong intensity in the visible light. So for this we have photosynthesis in the visible light. And so it is very much interesting the possibility that we have organisms on Earth, cyanobacteria, that can perform oxygenic photosynthesis using infrared light. This is because since a potential habitable exoplanet, of course, we can look at them only using space telescope or ground telescope. And we look at the atmosphere and we look at the life biosignature in the atmosphere. Uh, Oxygen and ozone is a very relatively easily it is easily to be it is easy to be detected in the atmosphere. So the possibility to look for sign of this kind of life form is also very important. And also, as we know on Earth, oxygenic photosynthetic organisms are green because they don't absorb light in the green. And so also surface biosignature could be a way to detect the signature of life on exoplanet. And so for this we are investigating since since M-star are very faint stars, habitable planets are very close to their star. And being so close means that they received also the flares and the UV radiation that the star is emitting. And so we did a laboratory simulation, let's say, of flares of stars, of M stars, and we used these two strains that we knew being able of red acclimation. So we made a comparison of cyanobacteria grown under white light or far red acclimated, and we used we used the UVC lamp at a certain distance. We didn't use the shaking because we want to enhance resistance by taking advantage of cell-cell shading. That is also a condition that we think could occur on an exoplanet. So there is no vortex that is keeping the cell in solution. And when we use the cyanobacteria grown under white light, we figure out that one strain was more resistant than the other. And 15 kilojoule, it's really a huge dose of UVC. And we noticed that the strain that was more resistant is also some interesting UV absorbent isopolysaccharides. And then when we used the strains that were cultivated under far red light, we saw that also the other strain was now more resistant. And also we noticed an increase of the presence of this UV absorbed pigment in the first strain, but also the appearance of these compounds that we think contributed to the increase of the UV resistance of these cyanobacteria. And then I want to now to be a little provocative, and I want to say something about the possibility of life on Europa and Enceladus, that are the two icy moons of our solar system that are target for astrobiology because under the surface they have liquid water. And liquid water is really, the presence of liquid water is rule number one to search for life. And so they are not receiving any light. A possible life here is not receiving any form of life because it is underground, let's say. But on Earth, they are in the hydrothermal vents. So these hydrothermal vents are appearing in the bottom of the ocean. There might be some geothermal radiation. And on Earth, there are some photosynthetic bacteria, non-oxygenic, so able to perform photosynthesis without the evolution of oxygen. And they are able to use this geothermal radiation that basically is an infrared radiation. So I'm a little provocative here because could be that life form capable of far-red acclimation could be also capable of oxygen production, or at least we can find photosynthesis also in this, let's say, icy moon of our in our solar system. So with or without the production of oxygen. And for this, we are also making some laboratory simulation of icy moon using these cyanobacteria, taking advantage of the fact that they are also very much desiccation tolerance. Because out of this icy moon we have some plumes, and mission are targeting these plumes and making a study in situ to see if in these plumes we can find some signature for life present under the surface. And so, since this rejection will be in space vacuum, doing this study, doing these studies using a desiccation tolerance cyanobacterium, it's also very much convenient. And all this, of course, has also some implication, not only for astrobiology, but also for space biotechnologies. So the possibility of having cyanobacteria so desiccation tolerance let us speculate that we could hard dry them on Earth. Maybe they can may have been genetically manipulated, so maybe they can have plasmid inside with sequence to produce something that could be needed to support human outpost on the moon or on Mars. They could travel so in space in the dried state. We know this for sure because we have the experiment that has been performed outside the ISS, and then we can think that we can rehydrate them once they are, for instance, on the moon. We have seen just yesterday that our astronaut Luca Parmitano will be the pilot of the Artemis 3 mission. And so once on the moon, we could rehydrate these cyanobacteria and have them to do what? First, it is important to cultivate cyanobacteria using in situ resources. So this is very easy to do. In the laboratory we use regoliths that are simulants of Moon and Mars soil, and you just need to add some water. Then, of course, you need light. In the case of desert cyanobacteria of the genus Crocosidiopsis, they are not able to perform nitrogen fixation, so we use urine as a source of nitrogen. This is also very convenient because urine, you go in the field of the bioregeneration system, so in utilizing also the waste from the crew. And then once that you have cultivated this cyanobacteria, you can use the biomass, for instance, to feed the bacteria that maybe at this stage are more suitable for space application. But being bacteria, they need to be fed with organics and they cannot do it by themselves. So it's very convenient to have a system to use in situ resources to feed the more complex, or more not complex, but heterotrophic bacteria. You can also fertilize the soil to grow plants. And in the future, we think to use these desert cyanobacteria for biotechnological applications, so to have them as, let's say, as cell factories to produce what could be needed by a human outpost, first on the moon and maybe later on Mars. And with this, I have to thank my astrobiology team at Vergata, my PhD student and postdoc, and funding, of course, from the Italian Space Agency, from the National Program of Research in Antarctica, Space It Up, the European Commission, and you for your kind attention. And I'm more than happy to take questions if you have.
Thank you so much for that wonderful talk. It's just fascinating topic, or a variety of topics, and you've left us on a
Yeah.
very provocative, thought-provoking note, which I really appreciate. So attendees in the room, please start entering questions into our Q&A box. You should be able to find the Q&A box at the bottom of your Zoom panel, or you can click the More button, which has the icon of a circle with three dots in it. If anybody has a question that's hard to type in, you can also raise your hand and the moderators, we can unmute you. Um, Well, I guess I'll get us started. I have a couple of questions, but I want to defer to the audience, so please go ahead and put yours in. To get us started, though, I guess you kind of ended on this note about comparing the cyanobacteria versus other organisms for bioreactors and kind of different applications for space biotechnology. Um, Great to see not only it could be a food source for various things, but it feeds in full circle with like using urine and kind of providing other important aspects that would help support like an off post. Is there, speaking specifically on the bioreactor, are there I know other organisms can do this, right? Like bacteria and things like that. Are there benefits, like the amount of mass that they can do, or like specific benefits that you think would make this a great system for like producing chemicals?
Yeah, and so they are not perfect. There are problems because they are slow growing compared to other cyanobacteria. So this could be, this is problem number one. But they are much more resistant to desiccation, to radiation. So this is important because you can transfer them in a dry state. This is very important. And they need also a lower attenuation of the surface condition because, of course, on the moon, on Mars, radiation is there, it's very intensive. And even if for some reason they get exposed, they can survive. So these are the benefits, the pro and contra of these things.
Great. So really robust, stable system,
Yeah,
but
but they're not
trade-off is
so fast.
growth.
Yeah, but for biomass production, yes, of course, we did it in the laboratory, but in large scale is a problem because they are not so fast growing. But of course, you may think about working on the division machinery and make them faster. So this is maybe something, a vision, but could be done, I think.
Oh, we're gonna need all the tools at our disposal. It's great to learn the nuances between them. Okay, we have a question came in from Thomas Boothby. What is known about how long these organisms can persist in the dry state? Is it decades? Do we know anything about the water content they survive at?
Yeah, I can say 1 decade, so 10 years for sure, because I have it in my laboratory and I keep storing them. So 10 years for sure. And then there is a very important project that was started already hmm, 5 or 6 years ago. That is the 500 years experiment that was started by Charles Cockle from the University of Edinburgh, and he prepared a lot of sample of Crocodiopsis dried and spores of Bacillus subtilis, and they were located at the London Museum, the historical museum, I think, in London, and all these vials are there, and every once in a while they are opened to check the survival. So in the future, in 500 years, we will know the record of survival because this is also very important, also for other issues. Traveling in space, lithopanspermia, how long microbes and spore can travel through space, we don't know.
Mhm, mhm. Yeah, thank you for that. Another question's come in from Taitra. Do these organisms survive radiation because they prevent DNA damage from occurring in the first place, or because they can repair the damage efficiently afterwards? Is there evidence that pigments or other protective molecules significantly reduce DNA damage, or do they mainly help the cells tolerate and repair?
Yeah, this is like it is known for the Deinococcus reducans. So having a very potent, yes, very, yeah, say yeah, potent system of defense against ROS, they reduce the amount of damage, of course. And they can also, but at a certain point, the damage occurs, and they can repair it because the extent, I mean, the amount of DNA damage depends to the dose. If the dose is very high, at the very end, you have the DNA damage is there. But being able, as I show, to protect, to avoid oxidative stress to protein, you can have the DNA repair protein working and repair and put a fragment, a DNA, a whole DNA from fragments. That is exactly what is known from the Inococcus redurans. And of course, if you have UV absorbing pigments, you can reduce the damage that are induced by UV radiation, but not the ionizing radiation, but the UVC radiation, yes.
Great, thank you.
So it is a balance between avoiding, limiting, and repair. So it is the three of them.
Yes, yes. Okay, perfect. And then another question's come in. Will markers, do cyanobacteria survive freezing when they are not in a dried state?
Freezing, not in a dried state. Um, Yes, usually it's better to dry and then put in the freezer because trehalose is also a cryoprotectant. But I put also pellet of cyanobacteria in the freezer without drying them before, and they survived. But it's better first to dry because you have the trehalose and sucrose accumulation, and trehalose in particular is a cryoprotectant, so it's much better if you do it in the dried state.
Yep, absolutely. Okay. And then we have a question from Kent Bradford. Clarifying, so just clarifying if you said that sufficient light energy can penetrate the rocks to support the growth of cyanobacteria below them.
Yeah, through, I mean, these kind of rocks are transparent, so they can let the light go in, but at the very end, the intensity of the solar light is as low as 0.1%. So they really like dim light. So when we cultivated them in the laboratory, we always use a very low intensity visible light because they really don't like high intensity of light.
Got it. Thank you. Okay. Are there additional questions you would like to put in? Or again, if you'd like to ask your question verbally, go ahead and raise your hand and we can try to unmute you. Well, I had a question similar to Chaitra's with repair versus prevent. And I think you showed some microscopy in dry conditions. I think it was like blue, green, kind of that nice slide you had. Were you looking at membrane damage? And do you see that also like repair over time, or is it if it's once it's damaged?
Yeah, when usually we look at the integrity of the membrane as a live dead cell. So
Mhm.
usually when we look at the membrane, if it is not if it is damaged, then the cell is dead.
Yeah. And you've looked at it like a single time point and then kind of counted dead and alive and then moved.
Yeah, usually we do the staining, and when you do the staining, then it's done because all these molecular probes then are not suitable for further cultivation, let's say. No, once that you do the staining, it's done.
Got it. Got it. Great. Thank you. Okay, well, we will wait a second to see if we get additional questions come in. While we wait for additional questions, I'm going to make a couple announcements, but I just want to start by thanking you again, Dr. Billy, for this wonderful presentation and sharing your work with us. We really appreciate your time and opportunity to learn about these really, cutting edge topics. Before people leave, I wanted to announce that we would like your feedback. I'm going to launch a short Zoom poll here on your screen, but again, continue to enter in any questions you have. We have a couple more minutes for Q&A. The Zoom poll will only take a minute or two to complete, and your responses will help us improve future events and understand topics and formats that are most useful for this community. We also hope you'll join us for future events. So this Anhydrobiosis Research Tools and Techniques webinar series is continuing. It is monthly. And the goal of this series, it's a mix of research seminars like we had today and technical training workshops. Um, all of the sessions have a goal of highlighting new scientific insights and practical methods to explore anhydrobiosis and support the desiccation tolerant community. But the technical training workshops are a little different, and we have one for next month. So the July talk will be a technical training workshop. These are, rather than seminars, they're aimed to kind of provide in detail training to support people to use techniques. Our presenter next month is Matt Stada, who's going to be presenting a workshop on exploring applications of RNA-seq data. He's going to provide cover a variety of software tools, and he's actually going to provide scripts for people to run the tools and modify them for their purposes. So we hope you'll continue joining us for this series that's a mix of seminars and technical training workshops. And as a note, this is monthly on the second Thursday at typically 8 a.m. Pacific, and please convert that to your time zone. So with that, I haven't seen any additional questions come in, so I will go ahead and launch our poll here. So the poll should be popping up on people's screen. And I will just say once again, thank you so much, Dr. Daniela Billi. We really appreciate the talk. This was super exciting to see. Um,
Okay, my pleasure. Bye.
thank you all. Bye-bye. And please, if you're filling out the survey, just wait a second. We'll keep the room open for a couple minutes. Oh, Dr. Billi, are we still here? We got one last question while people are filling out the survey, if you have time.
I'm here.
Wonderful. Thank you. So one more question came in from Olivia Carmo. So a follow-up on my question. She was wondering if there is a chance that the membranes could repair. Maybe we've kind of observed this in other systems. So we were wondering if the cultures could potentially be split and assayed at different time points, or if with the stain or method you're using to mark the membrane is perhaps that's unlikely, but we are wondering if maybe that might be interesting to
Yeah,
look.
of course, we cannot rule it out, of course. We cannot say no. But I think that we don't have the instrument to, to, to tell this, to prove this.
Ah, maybe it may be a future collaboration if somebody wants.
Yeah, yeah.
All right. Okay. Well, we're about at time, so I will say thank you again to our speaker, and feel free to roll out people, everyone, as you complete the survey.
Okay,
Bye-bye.
so I'm leaving. Bye bye.
Bye.